Dear students, after going through the salient features of the 24-hour-old chick embryo and 33-hour-old chick embryo, we are going to the features of the 48-hour chick embryo, that is one which is 2 days old. So, what are the characteristic features? We will see one by one. So, the first is the appearance of cranial flexure and torsion. So, the chick embryo undergoes two physical characteristics, two physical changes which can be named as flexure and torsion. So, to understand, we will go to some slides which will show you what flexion is and what torsion is. So, first one we will see what flexion is. So, here you can see what is happening here. Here the head of this person is bent forward at the neck. So, there is a transverse bending of the head forward at the neck region. That is what is called as flexion. So, in embryo, flexion means the bending of the body about a transverse axis. You can see this is a 33-hour-old chick embryo, and as time prolapses, you can see that this assumes a C shape because of a bending of the body. So, it undergoes a bending which will lead to a C shape of this embryo. That is what is called as flexion. Now, is this seen in all the embryos? No. This is a characteristic feature of embryos belonging to the group Amniota. So, who are amniotes? Reptiles, birds and mammals are considered to be amniotes. That means their embryos grow inside a watery bag which is called as the amnion. So, it is inside this space that the embryo grows. So, the space is limited. Whereas, in the case of anamniotes like fishes and amphibians, the embryos are growing in open water. So, there is no limitation of space. But in embryos, those who are growing inside the amniotic bag have got a limitation in space. So, to cope up with this limitation of space, what they do is they will undergo a bending so that accommodation is possible. Okay. So, flexion is a characteristic feature, or it is a necessary part in amniote development because of the limitation of space for development. In fishes and amphibians, there is no flexion since the development is in aquatic environment. So, this feature, this flexion, it can be seen in the embryos of amniotes, that is reptiles, birds and mammals. So, now we will go to the flexion in chick embryo. There are two flexures. This one, the anterior one is called as the cranial flexure and the posterior one is called as a cervical flexure. So, there are two flexures which can be seen in the chick embryo. So, what is cranial flexure? Whatever it is, the flexure occurs in a transverse axis. So, here you can see there is a bending of the neural tube at the anterior region. The neural tube is bent at the anterior region and it is bending through an axis which is passing through the midbrain. So, this is the axis through which the bend takes place. So, this axis, it is through this axis that bending occurs and this axis passes through the midbrain, and this occurs at the cephalic end of the notochord. So, here will be the notochord and at the level of the notochord this bending occurs. Notochord is seen ventral to the nerve cord, and at this level, at the cephalic end or at the anterior region of the notochord, or it is the level that marks the first flexure, that is called as the cranial flexure. So, it is a bending of neural tube in a transverse axis passing through midbrain at the cephalic end of the notochord. Now, what happens? This is the forebrain and this is the hindbrain. You can see now they are parallel to each other. This is the forebrain and hindbrain. Whereas here, this was forebrain and this was hindbrain. They were in a single line. Okay. They were in a single axis, but now because of flexion, the forebrain is now almost parallel to the hindbrain. So, this is called as the cranial flexure, and the second one is cervical flexure. Cervical flexure is the bending at the posterior region of the brain, and that takes place at the level of the rhombencephalon or the hindbrain. So, this is the bending which takes place at the level of the hindbrain, and that is called as a cervical flexure. So, because of these two flexures, the embryo it takes up a C shape. And remember, I told you flexion is a characteristic feature of embryos which are growing inside amnion. So, here in the whole diagram you can see this embryo which has assumed the C shape. Okay. So, this is called as flexion. Now, we will go to the second characteristic which is called as torsion. What is torsion? In human body, we will see what torsion is. Torsion is a condition where the person, without changing the position of his feet, he is turning his head and shoulders in looking backward. So, he is turning the anterior region of his body backwards without changing the position of the feet. That is what is called as torsion. Or with reference to the embryo, it is the twisting of the body of embryo along the anterior posterior axis. So, this is the level of torsion. It is at this region that the embryo undergoes a torsion. Okay, so it is the twisting of the body. And torsion is characteristic of embryos which are developing on large yolk mass. So, it is characteristic of embryos growing on a large yolk mass, and we will see what the reason is. So, before it undergoes flexion and torsion, as we saw in the 33-hour chick embryo, this was straight. The embryo was straight without any bend. So, like this man who is lying with his face down. Okay, he is lying with his face down. Just so was this embryo before flexion and torsion. It was also lying down. Okay. Now, so former to this phenomenon called as flexion and torsion, the embryo lies straight in its long axis. And where is it lying? It is lying upon the yolk. Just like this man's bed, the embryo is lying on a yolk bed until 33 hours of development. So, in this picture you can see the ventral side of this man is facing the bed, or the ventral side of this person's body is in contact with the bed. Just like that, in the initial stages, the ventral side of the embryo, this is the ventral side of the embryo, it was initially in contact with the yolk. Just like this man lying on the bed, the embryo was earlier assuming a straight position with its ventral side exactly in contact with the yolk. Now, what happens? The head undergoes flexion thereafter. Okay. And the head starts growing fastly and it bends downwards. Now, for further development, the underlying yolk is like a hindrance, the underlying large yolk mass will not allow further development if it does not undergo a rearrangement, because there is massive yolk lying beneath this embryo. So, without undergoing a readjustment, it cannot grow further. So, what happens? Thus, after the first flexion, before carrying out further flexion, the chick has to twist around on its side. So, full flexion is possible only if it undergoes a torsion, because there is yolk beneath. I will explain this. This man, he is lying with his face down. Now, he wants to assume this C-shaped position. This is the position in which the embryo has undergone torsion. Okay. So, this man has to undergo a C-shaped structure. Now, he has to adjust the space. So, before assuming this position, he will take a position where his body turns. Okay. He will turn and then assume this shape. So, there is an intermediate stage. This is the 33-hour chick embryo and this is the 48-hour chick embryo. In between this, there is a stage, that means he has to turn and then only he can assume this shape. Lying as such, he cannot undergo this torsion. He has to assume a turning, or he has to take a turning of his body. Okay. So, during that turn, the left side will be in contact with the bed and the right side will be up. You can see the right side is now coming upwards and the left side is in contact with the bed. Okay. So, this is all what is happening in the chick embryo also. That is, it starts torsion. The cephalic end of the embryo turns or twists in such a manner that the left side of the embryo comes in contact with the yolk. So, just like this man lies on bed, below is the yolk mass and the left side of the embryo is in contact with the yolk mass and the right side is now away from the yolk. Just like this man turned to this side with its right side up and the left side in contact with the bed. Just like that, the embryo now is in such a position that the left side comes to lie next to the yolk and the right side is away from the yolk. Okay. So, thus flexion and torsion change the orientation of the chick on yolk, and both these help in coping up with the problem of space limitation. So, flexion is carried out by amniote embryos who live inside the fluid contained bag called as the amnion, and torsion is characteristic of those embryos which are supplied by or which are lying on a large yolk mass. Okay. So, these are the most important characteristics of a 48-hour chick embryo, that is this flexion and torsion. Okay. Now, the next character, the second character. Second character is the formation of neuromeres. So, this is the neural tube which will form the prospective brain. Okay. So, this is the neural tube and this is the dorsal view of the neural tube. This is the prosencephalon, you can see the mesencephalon and the rhombencephalon. Okay. So, this is the hindbrain. So, this is forebrain, midbrain and hindbrain. Prosencephalon, mesencephalon and rhombencephalon. Now, what happens is that each region will develop some enlargements. Each region will develop enlargements which are called as neuromeres, or they in turn get divided into enlarged areas which are called as neuromeres. So, here in this diagram I will show you. This region is the prosencephalon. It is consisting of 3 segments. I will show you here. This is a dorsal view, this is a side view. So, this much part is the prosencephalon. Look, this much part is the prosencephalon, where you can see the first segment, the second segment and the third segment. Here it is marked as 1, 2 and 3. So, these 3 neuromeres constitute the prosencephalon. Now, 4 and 5, they constitute the mesencephalon. 6 to 11 constitute the rhombencephalon. So, these are in turn divisions of the brain. Okay. So, the forebrain or the prosencephalon consists of 3 neuromeres, 1, 2, 3. The mesencephalon consists of 2 neuromeres, they are 4 and 5, and the rhombencephalon consists of 6, that is 6, 7, 8, 9, 10 and 11. And they are all separated by constrictions. They are called as interneuromeric constrictions. Okay, so these constrictions can be called as interneuromeric constrictions, but when the development advances, these constrictions start disappearing. So, earlier the constrictions are very prominent, but when there is advancement of age, these constrictions start disappearing. But 2 constrictions will remain as such. This constriction between the prosencephalon and the mesencephalon, and this constriction between the mesencephalon and the rhombencephalon will remain. Okay. So, this constriction between the forebrain and the midbrain will remain even though other constrictions disappear, and this constriction between the midbrain and the hindbrain also will remain. Okay. So, those between 3 and 4 will remain, because 3 is the end of prosencephalon and 4 is the beginning of mesencephalon. So, this constriction will remain. Then that between 5 and 6 will also remain, because that is the boundary between the mesencephalon and the rhombencephalon. Okay. So, these are the neuromeres. Okay. Then, now the third character is the formation of secondary constrictions in the brain. That means the brain gets subdivided again. At 33 hour chick embryo, we saw the development of prosencephalon, mesencephalon and rhombencephalon. At 48 hour even by 38 hour, the brain it will divide into again different regions. So these are the three primary brain vesicles, which are prosencephalon, mesencephalon and rhombencephalon. They further subdivide. The prosencephalon subdivides into the telencephalon and the diencephalon. Mesencephalon remains undivided. The rhombencephalon divides into the metencephalon and the myelencephalon, which will continue as the spinal cord. Okay. So prosencephalon into telencephalon and diencephalon, mesencephalon remaining undivided, and rhombencephalon divides into metencephalon and myelencephalon. So that is the third characteristic of a chick embryo. Now the fourth one. Fourth one is the development of auditory vesicle. So this is the auditory vesicle here marked as the otic vesicle. So it marks the development of the ear. Okay, it is the sensory part of the ear. At 35 hours of development, a pair of thickenings appear in the ectoderm near the myelencephalon. Myelencephalon, just take a look here. Myelencephalon is a part of the hindbrain. So at the level of the myelencephalon, thickening appears in the ectoderm here. So corresponding to this region, this is the ectoderm covering the embryo, and here a thickening appears on the other side also, the same happens. So they form a pair of auditory vesicles or auditory placards, you can call them, auditory placards. This occurs at 35 hours. By 38th hour, these placards, they become depressed to form pits. So earlier they were thickenings, and these thickenings further depress, and they form pits which are called as the auditory pits. And these pits, or the opening of these pits, become narrowed, and finally they close and they become the auditory vesicles. Okay. So initially you can find a thickening which then forms a pit, which then gets closed and which forms the auditory vesicle, which begins the marking of ear development. So this is all about otic vesicle. So by 48th hour you will be seeing the auditory vesicle formed. Now next is the development of optic vesicles. It had already started at 33 hours. So this is the brain. This is the forebrain, the midbrain, the hindbrain. Okay, so this is a brain, this is the brain, this is the brain, where this is forebrain, midbrain and hindbrain. Okay. This is the head of the embryo, the ectoderm, this is the epidermal ectoderm, this is the neuroectoderm which forms the boundary of the brain. Okay. And we already saw that from prosencephalon, two evaginations arise. They can be called as optic vesicles. So these two are the optic vesicles. Okay. So this much we had discussed, or we had seen until 33 hours of development. Now what happens by the time the embryo reaches 48 hours? So this part is enlarged here. Okay, so I have taken only one side which is enlarged here. So this is the optic vesicle. In this diagram, what is the optic vesicle doing? It is growing towards the ectoderm. This is the ectoderm. So the optic vesicle grows towards the head ectoderm. Okay. And then what happens? It will invaginate. So this is now, you can see it is invaginating. Okay. And invaginating to form a cup which is called as the optic cup. So this is the optic cup which further deepens. And the optic cup has got a stalk which is called as the optic stalk. So this is the optic stalk. Here this is the optic stalk actually. This is the optic stalk. So since this is an enlarged view of this part, this can be considered as the optic stalk. So this is optic vesicle, the optic cup, and the further deepened optic cup which will develop into the eye, and the optic stalk. Now the part of this ectoderm which is now in contact with the optic vesicle undergoes a thickening, that is called as the lens placard. So this is a lens placard we saw in frog development also. This also invaginates. So the lens placard is formed from the ectoderm, it now invaginates. Okay, opposite to the optic cup. and you can now call it as a lens vesicle. It's called a lens vesicle that opens to outside through a small aperture. So, this is the aperture through which the lens vesicle opens outside. It further deepens. You can see in the next diagram what's happening. The optic cup is fully formed and this lens placard has pinched off. It has been pinched off from this ectoderm. This deepens, forms a vesicle, and these two areas fuse together and it is pinched off as a vesicle, and that is called as the lens vesicle. So, the same events which we saw in the eye development of frog. Okay. So, this is optic vesicle which deepens to form the optic cup. This is the lens placard which is invaginating, deepening, and finally getting pinched off as a vesicle, and which is contained inside the optic cup. Okay. This is not complete by 48th hour. Even you look at the embryo at 48th hour, you can see the pinching off is not complete. At 48th hour also you can see it is opening to outside. This is a later stage of development where it is pinched off. Okay. So, this stage will be attained only after 48 hours. At 48 hours you can see it only deepening, still maintaining a connection to outside. Okay. So, that is all regarding the development of the optic vesicles. Now, we will go to the somites. What were somites? At 21st hour, the first pair of somite was formed. Then, at 24th hour, we found that 4 pairs of somites are formed. At 33 hour, 13 pairs, and at 48 hour, there will be 28 pairs of somites. Okay. So, 28 pairs of somites will be formed. So, differentiation of somites into 28 pairs. The stage is attained at the 48th hour. Now, we will go to the next one, that is the completion of vitelline circulation or extra embryonic circulation. In 33-hour chick embryo, it was mentioned that the omphalomesenteric veins they develop. This is the omphalomesenteric vein which is collecting blood from the yolk sac. So, it had started developing in the 38-hour chick embryo itself from the sinus terminalis. Okay. Then, so this is the omphalomesenteric vein which is connected to the vitelline veins which will ramify the yolk sac. So, the blood from the yolk sac is collected by the vitelline veins which goes to the main omphalomesenteric vein which is emptied to the heart. And by 48 hour, the vitelline veins are fully connected to this omphalomesenteric vein and the circulation starts full fledged. By 40th hour itself, there is full connection established between the omphalomesenteric vein and the small vitelline veins, and this vitelline circulation starts. Now, what about the vitelline arteries? The vitelline arteries, they supply blood to the yolk sac, they are developing from the dorsal aorta. So, by this the vitelline circulation almost begins. This is what is called as the vitelline circulation, that means circulation between the yolk sac and the heart. The veins collect blood from the yolk sac and empty it to the heart. The arteries, they bring blood from the heart towards the yolk sac. So, these are the vitelline arteries and these are the vitelline veins. So, this is called as extraembryonic circulation or vitelline circulation. The next is intraembryonic circulation. At 33 hour, we saw the development of the ventral aorta, the dorsal aorta and the development of aortic arches. So, it was mentioned that at 38th hour, there will only be one pair of aortic arch. But as time progresses, there will be development of more aortic arches. Now, this intraembryonic circulation, that is the circulatory system existing between the embryo, the proper embryo and the heart, that is called as intraembryonic circulation, whereas that between the heart and the yolk sac is called vitelline circulation, but the circulation between the heart and the embryo proper is called as intraembryonic circulation. So, it will be having a arterial system and a venous system. That means a system that brings blood to the heart and a system that takes blood away from the heart. The system that brings blood to the heart can be called as efferent, and the system that takes away blood from the heart can be called as afferent. Okay. So, we will go to efferent circulation. Efferent means the blood is taken away, the blood is taken from the heart. One which comes to the heart is called afferent, and the blood drained is called as efferent. So, we go to efferent first, that means the blood which is coming out. This is ventral aorta, it bifurcates into two. So, this is only a side view, the same thing happens on the other side also. So, this is the ventral aorta. It will curve around the gut and develop into the dorsal aorta, and the ventral aorta and dorsal aorta are connected by the aortic arches. The same thing happens on the other side also. This is only a side view. So, these are the aortic arches, and you can see from the dorsal aorta branches are given off to various parts of the body. The ventral aorta also gives off branches to various parts of the body. So, in concise, in short, we can say the ventral aortae and the dorsal aortae, they form the main efferent conducting vessels of embryonic circulation. That means, they are the blood vessels which start circulating blood to other parts of the body. So, they can be considered to be the main efferent conducting vessels of embryonic circulation. That means, they give off branches to various parts of the body. Then we go to afferent circulation. That means, the circulatory system that brings blood to the heart. It consists of two pairs of cardinal veins. This is the anterior cardinal vein, which is collecting blood from the cephalic end of the embryo, and this is the posterior cardinal vein, which is collecting blood from the posterior parts of the embryo. They together form the common cardinal vein. The same phenomenon can be seen on the other side of the embryo also. This is a paired structure or a paired phenomenon, and these cardinal veins, they open to the sinoatrial end of the heart. So, this is the heart, where this is called as the sinus venosus, this is the atrium, this is the ventricle, and this is the ventral aorta. Okay. Sinus venosus, atrium, ventricle, and ventral aorta. Ventral aorta initiates the efferent circulation, and it is into the sinus venosus that the afferent veins bring blood. It consists of the anterior cardinal vein that brings blood from the cephalic end of the body, and the posterior cardinal vein which brings blood from the caudal end of the body. They join to form the common cardinal vein, and there will be one more cardinal vein from the other side also, forming the same type of connections. And the next feature is the formation of aortic arches. Aortic arches are nothing but curved blood vessels connecting the ventral aortic rod with the dorsal aortic root, and at 48 hour you can see two pairs of aortic arches. So, this is a much advanced diagram. At 33 hour there will be only one aortic arch, and at 48 hour you can see two pairs of aortic arches in the 48 hour embryo. So, by 48th hour the heart starts pulsating, the heart starts beating, and commencement of blood circulation is also there. That means, blood circulation is almost completely formed, and it establishes itself into two parts: the intraembryonic circulation between the embryo proper and the heart, and the vitelline circulation between the heart and the yolk sac. So, these are the two parts of the blood circulatory system. Now, we will go to the heart, the structure of the heart itself. In 33 hour we saw, or by 25th hour we can see there are two heart tubes or endocardial tubes which fuse to form a common tube which forms the heart. So, by 33rd hour you can see a bending of the heart to the right side. So, this much changes we had seen by 33 hours. So, the mid region of the heart is dilated and bent to the right. Even after this bending, the heart will continue to grow. Even though the heart is fixed at both ends, it will continue to grow. So, what happens? It is fixed at both ends, but the growth continues. So, this leads to the twisting of the heart into an S-shaped spiral tube to accommodate itself. So, this is so it is growing, but it is fixed at both ends. So, this its growth or the grown tissues has to be accommodated. So, for that it will undergo a twisting in the form of an S-shaped spiral tube which has got 3 parts. This posterior part, this is the sinus venosus of the heart. This is the sinus venosus which will receive the veins. The veins empty into sinus venosus. It opens to the atrium. The atrium opens to the ventricle, and ventricle is that part which forms the U-shaped bend, and the ventricle narrows down into the trunk. The truncus arteriosus. Earlier this split into 2 ventral aortae. Now it becomes the truncus arteriosus which will split into the ventral aortae. So, these are the changes which you can see in the development of the heart and the shape of the heart also as it is being formed. At 44 hours, the rhythmic heartbeat is re-established. So, what you can see is the development of parts of heart, that is the sinus venosus, the atrium, the ventricle which opens to the truncus. So, these are the regions. Now, the next feature in a 48 hour chick embryo is the formation of visceral clefts. What do you mean by visceral clefts? So, we come to that. Suppose here is the mouth. Mouth opens to buccal cavity opens to the pharynx. So, this is the pharynx. So, this passage is the pharynx. The pharynx will lead to the esophagus, you know. This is how the digestive tract is in. So, mouth leads to buccal cavity which leads to the pharynx. So, this space is the pharynx. So, this region can be considered as the wall of the pharynx. You can see the wall of the pharynx on either side. So, these two are the lateral walls of the pharynx. So, this is the anterior part of the digestive system. This is the cavity of the pharynx, and this is the wall of the pharynx, and this is again the pharyngeal wall, the lateral walls of the pharynx. From the lateral walls of the pharynx, you can see pouches arising. Pouches. These pouches are called as visceral pouches. So, the wall of the pharynx, the lateral wall of the pharynx is giving off invaginations which are called as pharyngeal pouches. They are paired structures, one on this side and the other on this side. The second pharyngeal pouch, the third pharyngeal pouch, and the fourth pharyngeal pouch. So, by 48 hours there will be 4 pairs of visceral pouches. Where are they growing to? They are growing towards the ectoderm. This is the ectoderm. This is the ectoderm or the outer surface of the embryo. So, this is the outer surface of the embryo, and these pouches, they are growing towards the ectoderm. At the same time, the ectoderm also forms invaginations. The ectoderm is also having invaginations. You can see one invagination growing to this direction, second invagination growing to this direction, third invagination growing to this direction, and fourth invagination growing to this direction. So, they are growing towards the pharyngeal pouches, and these invaginations of the ectoderm are called as pharyngeal grooves. The first pharyngeal groove, the second pharyngeal groove, the third pharyngeal groove, and the fourth pharyngeal grooves. So, pharyngeal evaginations or pouches are called as pharyngeal pouches, whereas the ectodermal invaginations are called as pharyngeal grooves. Okay. Now, they are growing towards each other. They can meet, and this forms a space here. A slit will be formed. If they meet and the tissue between them disappear, here a full passage will be formed. It is called as a visceral cleft. This is well formed in aquatic organisms like fishes, amphibians, etc. But even though it is there in birds, reptiles and mammals, they soon disappear. They soon get closed because they are not aquatic organisms, but they are terrestrial organisms. But in fishes and amphibians, the clefts will remain as clefts themselves because they breathe through gills. It is in the clefts that the gills develop. But in chick embryo, this is not needed. So we can see there will be formation of pharyngeal pouches as well as the formation of pharyngeal grooves. Of course, the first one will meet and form an aperture here, but it will not develop into a gill chamber because it is not aquatic. But this will, the first one in a chick embryo will contribute to the development of auditory canal. In humans, this first one contributes to the development of eustachian tube of the ear. In chick embryo also, this contributes to the development of the auditory canal. Whereas others are just temporary. So this is also a characteristic feature of 48-hour chick embryo. So the visceral clefts are only transitory. And what does this imply? That means there is a theory which is called as ontogeny recapitulates phylogeny. That means the developmental history of an organism reflects its evolutionary history. So this is the developmental biology of chick, but it almost resembles the development of a fish. Okay, so that is why it is said that ontogeny recapitulates phylogeny. That means the development of fish embryo to a particular stage, there are some similarities in the development of fish embryo, amphibian embryo, reptilian embryo, chick embryo and mammalian embryo. That is what it indicates. But they will soon get closed. They won't develop into functional gills because they are terrestrial organisms once they become adults. But this type of pharyngeal pouch formation is there during development. These are the pharyngeal pouches. These are the pharyngeal grooves. Pharyngeal pouch can also be called as visceral pouch. This can also be called as a visceral groove. Now, this will be the first cleft. This will be the second cleft if they grow towards each other. And the space in between, this one is called as the visceral arch. So, the mesodermal portion between the first cleft and the second cleft is the arch, or the second cleft and the third cleft. This is the next arch. So, they are called as visceral arches. The mesodermal portion between visceral clefts differentiate into visceral arches. This is an enlarged diagram of this portion. So, this is a one-sided diagram where you can see pharyngeal invagination of pharyngeal pouch, the pharyngeal cleft, that is the ectodermal invagination, second one, third one, fourth one, like that. So, this part has been enlarged like this. Okay. This is pharyngeal pouch, pharyngeal cleft, second pharyngeal pouch, second pharyngeal cleft, third pharyngeal pouch, third pharyngeal not cleft, groove, fourth pharyngeal pouch and fourth pharyngeal groove. Next is the development of extra-embryonic membranes. What are extra-embryonic membranes? They are a set of membranes covering the embryos of amniotes. Who are amniotes? Reptiles, birds and mammals. There are 4 membranes which cover the embryo of amniotes. They are called as the first one, this one. This is the embryo. This is the embryo. The first covering, it's called chorion. Next is this one, which is forming a fluid filled bag around the embryo. It is called as amnion. The third is allantois and the fourth is yolk sac, which provides nourishment to the growing embryo. So, this can be called as the extra-embryonic membranes. They are derived from the embryonic tissue itself, but they do not form a part of the proper body parts of the embryo. They do not form the proper embryo. This is the embryo proper, but these are all accessory structures which can be seen during embryonic development only. So, the first one is chorion, second one is amnion, which forms a bag around the embryo, third one is allantois and fourth is yolk sac. So, that can also be seen during the 48-hour embryo, the formation of extra-embryonic membranes. And among all the 4, the yolk sac is the first to develop. It is developing from the splagnoplure. Splagnoplure is the mesoderm which is close to endoderm. So, the first to develop is the yolk sac, the chorion and the amnion. They develop from the somatoplure, that means the part of the mesoderm which is in contact with the ectoderm, that is called somatoplure. So, the amnion and chorion are developing from the somatoplure. Allantois is the last one to be formed. It is formed on day 3. This is another characteristic feature of a 48-hour chick embryo. Then next is the development of excretory system. We saw this diagram in 24-hour chick embryo where the mesoderm differentiated segmentally into 3 regions. This is called as the dorsal mesoderm, this is called as the intermediate mesoderm, and this is called as the lateral mesoderm, which split into somatic layer and the splanchnic layer. All these features were discussed in 24-hour chick embryo. So, this is what happens to the mesoderm in each segment of the embryo's body, that is, this is the dorsal mesoderm, this is the intermediate mesoderm, and this is the lateral mesoderm. Now, what happens to this intermediate mesoderm? So, this is a structure which is segmentally arranged. The dorsal ectoderm, it will give rise to the somite. Okay. Now, what happens to the intermediate mesoderm? Intermediate mesoderm is also called as the nephrotomic plate. In each segment of the body, the nephrotomic plate, it will give rise to a structure here. This is called as the pronephros. So, they are developing from the intermediate mesoderm. Remember, this is a transverse section through the embryo. This is a full embryo. So, imagine this is a section. These two are entirely different diagrams. You can see from entirely different planes. So, do not confuse. This is the transverse section where you can see the intermediate mesoderm. You have to imagine a full embryo which will be having such structure in each segment. So, the intermediate mesoderm gives rise to such structures. The structures which are seen on the excretory structures which are seen on the anterior side of the body are called as the pronephros, but they are non-functional. They do not have a tube to take up excretory waste. By 33 hours, the nephrotomic plates, they start differentiating into non-functional pronephros. They do not take up the full function. Afterwards, and the pronephros, they will give rise to a kidney at the level of the 5th to 16th somite. You can see here will be the somites, and at the level of the 5th to the 16th somite, you can see this pronephros will give rise to a kidney. They all together will give rise to a kidney, but it is non-functional. By the 3rd day, this starts to disintegrate, and from the posterior nephrotomes, here also there were nephrotomes, and they give rise to a kidney which is called as a mesonephric kidney. So, by the 3rd day, you can see a mesonephric kidney developing. In adult, it is the metanephric kidney, but in the embryonic stage, there will be a mesonephric kidney. These are the pronephros which are non-functional. So, in short, the excretory function is taken up by such segmentally arranged structures called as the nephros. The anterior ones are the pronephros, the middle ones are the mesonephros, and from the posterior region. In adult hen, the posterior tubules, they give rise to the kidney. This is called as the metanephric kidney, but in embryonic stages you can see the mesonephric kidney taking up function. This is all about the excretion. And the last characteristic of a chick embryo, which at 48th hour of development, is the formation of liver bud as an outgrowth of the foregut. So, this is the gut. This is the whole embryo, this is the gut, and as an outgrowth of the gut, a liver bud develops and liver starts to develop. This is only a representative diagram where you can see from the gut the liver develops as an outgrowth. One more structure develops in the 48th hour embryo, that is the thyroid gland. It also appears as an outgrowth of the foregut. So, these are the major characteristics of a 48-hour chick embryo. So, we will again go to the characteristic features in a nutshell, in concise. First one is the appearance of flexion, that means assuming a C shape, torsion, twisting of the body, then formation of neuromeres in the brain, the division of the brain into different regions, the brain vesicles, then the development of the auditory vesicle, the development of the optic vesicle, optic cup and the lens primordium. Then there will be formation of somites. You can see how many pairs of somites, 28 pairs of somites will be there in the 48th hour. So, these are the somites. Then the development of circulatory system, both extraembryonic circulation and intraembryonic circulation starts. In the intraembryonic circulation, there is efferent circulation carried out by the ventral and dorsal aorta, and there is afferent circulation carried out by the cardinal veins. So, there will be establishment of intraembryonic and extraembryonic circulation. Then the heart undergoes twisting and develops sinus venosus, atrium, ventricle and truncus. Then the formation of visceral pouches and visceral arches in between. Though the first cleft will be found, it is only a transient or a temporary phenomenon. Then extraembryonic membranes, chorion, amnion, yolk sac and allantois start to develop. Yolk sac is the first formed. Taking up of excretory function by the mesonephric kidney, which is formed from the intermediate mesoderm. The nephrodes are structures which are segmentally arranged. They are formed from the intermediate mesoderm in each segment. They take up the excretory function. A mesonephric kidney appears, and the formation of liver and thyroid as outgrowths of the gut. So, this is liver. Here also you can see the liver and thyroid as gut outgrowths. So, these are characteristics of the 48-hour chick embryo. Thank you.